Hello everyone and welcome to the session 3 of NPTEL course in pharmacognosy and phytochemistry. By now we know what is pharmacognosy and phytochemistry, what is the scope of the subject and what are different drugs of natural origin that we saw in the previous session. Now in this session we are going to go a little step ahead in understanding what compounds does this drugs contain. So in this session we are going to learn plant metabolites and more predominantly we are going to classify them as primary and secondary metabolites, and we are going to understand their biosynthetic precursor. So what are these plant metabolites and what are primary metabolites? Now whenever a plant thrives in an environment, there are numerous compounds which are produced of it, and this process that of producing compound is called as metabolism, and that is the reason they are called as metabolites. Now some of this metabolism happens to benefit the plant. The compounds coming out of it are called as primary and secondary metabolites. Now if you go to see primary metabolites are vital or essential metabolites which are required for the growth, development and reproduction of the plants. Now if you see molecules such as sugars, proteins, lipids, nucleic acid and vitamins, by now understanding the biochemistry of plants, we know that they are very vital. So whenever there is a sunlight, the first thing that the plant produces is it is going to take carbon dioxide and water, and through a process of photosynthesis it will give rise to sugars. Now these sugars are the main source of energy to the plant which will help in developing the plant. Now these sugars will further undergo numerous pathway to develop other metabolites which are essential for its survival. So from sugar pathways we will get our proteins, we will get our lipids. Now the plant genetic material is very vital, so we have our nucleic acids and also vitamins which are cofactors to numeric biosynthetic processes. So all these compounds put together are called as primary metabolites. Now there are some compounds or some products of metabolism which are not essential, but they attribute a particular quality to the plant. This quality can be a defensive quality, or this quality can help them prevent from a disease. Not only that, in some cases these principles also help us attract beautiful insects. Now these compounds or this set of metabolites are called as secondary metabolites. So secondary metabolites are little distinct metabolites which are further derived from primary metabolites, and their function is to prevent the plant from disease, prevent the plant from infection, communicate with other plants, help the plant undertake stress, be it environmental condition, be it pH of soil, be it drought, be it rain, be it any stressor. Now this stress metabolites will help plant to thrive in this particular condition. Now because these plants are, or because these metabolites are produced to repel insects or in certain cases to attract insects, they are defensive or protective in functions as compared to primary metabolites. These compounds are found to have more pharmacological effect on animals as well as on us humans. And as a result, when you are searching for bioactive molecules such as drugs, the secondary metabolites are predominantly important to us. Some of these metabolites render antibacterial properties, essentially the phenols and the tannins. Some of them are cardiotonic. When we know digitalis, it produces something like a digitoxin which is a cardioactive compound, and there are certain substances which plants will produce which will cause hallucinogenic effect. Now you know something like your charas, ganja, they are all plant based metabolites, but you know they have what is called as CNS stimulating effect. So that is also one way of plant to be defensive, and in order to tell the animal that you should not consume this. Some substances are intensely bitter, and as a result they will deter the animal from consuming it. These compounds include something like your steroids. So numerous compounds such as flavonoids, terpenes, antibiotics, alkaloids in fungus, aflatoxins are secondary metabolites that are produced, which will help give all these functions to the organism. Now, like I said, from pharmacological perspective or from medicinal perspective, the secondary metabolites are very vital to us. Now, where are these metabolites located? Suppose if I want to explore a medicine, I want to take the secondary metabolites out from the plant, where do I take it? Now, the secondary metabolites have a little different location depending upon their polarity. So, depending upon their polarity, you can divide the secondary metabolites into two categories. One is hydrophobic, that is very lipophilic or lipid like molecule. One is hydrophilic, they are more water soluble or water loving compounds. Now, if you see hydrophobic, that is waxy or lipid like compounds, you will see them especially in the outer coatings. So, if you see your leaf does not get moist during the rainy season because they have that waxy coating. So, plant waxes, terpenoidal or a very hydrophobic compounds can be located in the outer cuticle, that is outer lining of the epidermis. They can be located on trichomes. So, if you know cotton is an example of very long trichome which is present, and very few of us know that this cotton is actually water repelling. But if you somehow remove that waxy coating from cotton by a process called as defatting, only then the cotton becomes absorbent. So, you will have your fats which are coating the cotton, preventing it from soaking in the rain, but when we want to use it, we will defat it and make it more absorbent. So, the fat is there on the trichomes. Similarly, you might have some terpenes which are located on trichomes. Say for example, your charas ganja is a kind of resin. It might contain terpenoidal compounds, it might contain phenylpropenoidal compounds. The mixture of all of this are located inside the glandular trichomes which are there on this cannabis plant. So, you can just touch it and you will get this transferred to your hand. Now, in some cases they might occur in the resin duct. There are something which we discussed in the previous session which we called it as resin. Say for example, asafoetida or even pine resin, they are located in resiniferous ducts. So, that might contain terpenes, flavonoids, phenols or any derivatives. Not only that, there are certain cells inside the plant which are called as oil cells. They specifically localize oil droplets, and these oil droplets are stored till you cut it. So, these are called as oil cells which are located in your chilies or in your gingers, and sometimes instead of a cell it might be a huge pipe or huge duct. So, you might have or you might come across oil ducts. If you see umbelliferous fruits such as fennel, coriander, the oil is located not in cells but in form of ducts. We call it as vitae. So, you might come across some phenylpropenoids or terpenoidal compounds which are present in oil ducts of such fruits. Now, there are certain compounds called as quinones which are responsible for your electron transport chain, and they may be located in your plastid membranes. So, these are few locations where you will find your lipophilic or hydrophobic molecules. Coming to your hydrophilic or water soluble molecules, you will see the leaf mesophyll region or parenchymatous region, especially the vacuoles. Those are the places where you have abundant sap or water coming out. So, there in the vacuoles you will come across compounds such as saponins, glycosides which are nothing but molecules to which sugars have been attached, anthocyanins which are beautiful pigments present in your flowers responsible for the color of certain flowers such as your periwinkle. Now, some of them may be there in the lactiferous duct from where the latex oozes out. An example of that is your opium or in some cases your lactiferous ducts of papaya will contain numerous proteins derivatives and so on. Your cell walls, especially if you see the trees, the outer wall of the trees, especially the stem, is brown in color, and that brown in color or that brown color is due to deposition of compounds called as tannins. So these are few places where you will find your hydrophilic metabolites. Now coming to the biosynthesis of secondary metabolites, we have your biosynthetic pathway, especially in plants, something which starts from something as simple as photosynthesis. So what happens in your photosynthesis is you have your CO2 plus water just taken up by plant in presence of sunlight, and this process is called as photosynthesis, whereby this add up to make your sugar. This simple sugar includes your glucose, but later on there are numerous pathways called as your shunt pathways which help in developing other sugars as well. So you know you have your ribose, pseudoheptulose, pentoses and so on. So from photosynthesis you will get your glucose, and via different shunt pathways you will get other saccharides also. Now this saccharides right from glucose to others will start polymerizing, that is adding them. Now why does plant does this is a process called as osmosis. So if too many sugars accumulate in the plant, it will have a very high osmotic pressure. So in order to decrease it, the plant kind of polymerizes it and precipitates it, whereby that osmotic pressure is no longer exerted in that manner, and that's where you get your oligosaccharides and polysaccharides. So you will have your starches as polymers of glucose, you have your cellulose as polymers of glucose, you have your different oligosaccharides, polysaccharides, as well as some of the sugars get converted into your polyols. So all of this are coming from your sugar pathway, that is your photosynthesis pathway. Not only that, if you take your glucose, this glucose will further undergo Embden-Meyerhof pathway, and then it will get converted into your pyruvic acid. So if you recollect, your Embden-Meyerhof pathway was a pathway wherein your glucose gets converted into fructose, and thereafter you split it into your C3 compounds, and the main or the end product of your Embden-Meyerhof pathway is your pyruvate. Now this pyruvate further gets converted into acetyl-CoA. Your pyruvate and acetyl-CoA are important byproducts of your Embden-Meyerhof pathway, and later on you will see that your acetyl-CoA enters your citric acid pathway. Now before that, acetyl-CoA may undergo addition or polymerization to give you other secondary metabolites. This includes your fatty acids, waxes, terpenes, saponins and quinones. So they all come from this particular pathway, or they kind of diverge from this particular pathway. Now once your acetyl-CoA goes into citric acid pathway, in your citric acid pathway you have your malic, succinic, fumaric acid and so on. Now this acids or this organic acids will undergo a reaction called as transamination reaction, and this transamination reaction will convert this organic acids into amino acids. Now these amino acids are essential, and these amino acids can further be converted into next set of secondary metabolites. This include your alkaloids as well as antibiotics. So you will see that your basic pathways can just further extend and manipulate in themselves to give secondary metabolite compounds. Now let's see a few building blocks of secondary metabolites, and let's focus them one by one. Now when you say your secondary metabolites, they include compounds, and in this case we will be talking about organic compounds. Now when I am talking about organic compounds, it is made by addition of numerous subunits. Like you are building a house brick by brick, a molecule is built by brick by brick. Now each of this brick is called as building block, and depending upon how I want my structure to be, I can choose my building block. So that is how your plant decides what it is to be chosen to in order to get a particular secondary metabolite. Now starting with most simplest compound, the simplest compound or the simplest building block is C1. So imagine a case when I want to add just one carbon. Now this carbon generally occurs in form of methyl CH3, and this methyl is given by an amino acid called as methionine. So suppose if you know your amino acid methionine, you know in this case what happens is methionine is an amino acid wherein your methyl group is attached to a sulfur. Now a adenosine is something which is genesized or which is biosynthesized from numerous pathways, but the basic molecule here is ATP, adenosine triphosphate. ATP fuses with your methionine to give what is called as S-adenosyl methionine. Now what happens is this group carries a slight charge, and as a result your methionine groups can easily be transferred. So SAM or S-adenosyl methionine is an excellent methylating agent. Now this generally happens in the cytoplasm, but the movement of SAM is done by SAM transporters which help move S-adenosyl methionine to different organelles. This includes your mitochondria, chloroplast and numerous places wherever you want to carry the methylation reaction. Now where does methylation happen? The substrate for methylation can be a O, that is will give rise to OCH3. The substrate can be a N, which will give rise to NCH3, or in some cases or very rarely C, which will give rise to CCH3. Okay. So this is very rare, but first two cases are very common. Now looking at the examples, so suppose if you see a molecule such as ferulic acid, now I am focusing on CH3. Why? Because I am seeing this building block. So whenever you see a natural product with a methyl substitution of the nature OCH3, NCH3, can you spot it? Here it is. So in your ferulic acid you can see a OCH3, and this CH3 group has been contributed by S-adenosyl methionine. Similarly in your eugenol, which is there in your eucalyptus, again you can see a OCH3 of this CH3 group has been contributed. So whenever you want to methylate anything or whenever you want to add an carbon, this is something which will help you do it, and that is your S-adenosyl methionine. Now going to complex molecules such as podophyllitoxin. In this case also you will see OCH3, OCH3, OCH3. So there is 1, 2, 3 molecules wherein your SAM has helped. Now let me tell you, it's not 3, it's specifically 4. The fourth place is this methyl. So it is not just OCH3, it could be something like OCH2O also in some cases or some few cases where your SAM also helps in. So this is a fourth case where your S-adenosyl methionine has helped add you one carbon. So whenever in plants you see a unique isolated single carbon moiety, that has been contributed by your S-adenosyl methionine. Now let's move on to the second building block, and that is a little bigger brick. Instead of C1 we have a C2. Now C2 comes from 2 carbon unit. So where does this 2 carbon unit, or what is the precursor of this 2 carbon unit? The precursor of 2 carbon unit is generally an acetyl CoA. This is your C1, C2. But if you see in terms of reactivity, your acetyl CoA is little less reactive. So what happens in plant is this acetyl CoA in presence of biotin, carbon dioxide and ATP, as well as an enzyme called as acetyl CoA carboxylase, will get converted into malonyl CoA. Malonyl CoA is a more reactive substrate as compared to acetyl CoA, but will still give you the same 2 carbon addition. What happens is this particular group gets eliminated when it goes on adding. Now you imagine something like a big fatty acid, which is a even chain fatty acid. Now stearic acid is basically a C18 fatty acid. So I can just break it as C2. So I should have 9 cuts ideally. So this is C2, 2, 3, 4, 5, 6, 7, 8, and this is the 9th one. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 C2, so 9 into 2 carbons is equal to 18 carbons is stearic acid. So similarly you have your lauric acid, you have your palmitic acid, C12, C14, even C22, C30. You can see the number of carbons and you can just go on multiplying. You will know how much amount of acetyl CoA are going to be required for that particular fatty acid just by dividing it by 2. So if it is C30, you are going to require 15 number of acetyl CoA. Suppose it's a C20, you are going to require 10 number of acetyl CoA in order to produce it. So what happens here is it gets converted into malonyl CoA, gets added, but when every time it gets added, this carboxylate is eliminated. So you might have fatty acids, you might have polyketides, and you might have phenolics. Now you might ask, what are polyketides? Now when I say acetyl CoA, I can also represent it in this manner. What is generally happening in case of fatty acid synthesis is all my ketone groups here are getting reduced. So imagine a case where they don't get reduced at all. You get compounds which are called as polyketides. So polyketides are very common in marine scenario. Polyketides often fold to give you compounds called as quinones. Sometimes polyketides condense to give you phenolics, and this phenolics, a good example of that is your one of the ring in flavonoid is coming from a folding of acetyl CoA. So you can give rise to fatty acids, polyketides or phenolics with help of acetyl CoA, that is C2 as building block, but most simple example is your fatty acids. So now the next set of building block is we have C5 building block. Now if you go to see the building blocks in between, C3 and C4, they are not as abundant, but C3 generally comes from pyruvate or glyceraldehyde 3-phosphate, whereas C4 generally comes from erythrose or succinic acid units. The abundant one is a C5 unit, which leads to formation of steroids, terpenoids and many other compounds such as essential oils. Now if you go to see C5 unit, C5 unit consists of a monomer which is referred to as isoprene. Now isoprene is a C5 monomer and it has its genesis from two molecules or two key biosynthetic intermediates. This include mevalonic acid as well as 1-deoxy-dexhydrolose 5-phosphate. Now mevalonic acid as well as deoxyxylulose phosphate come from different pathway, whereas your mevalonic acid comes from your acetate mevalonate pathway. Your deoxy-dexhydrolose phosphate comes from methyl erythritol phosphate pathway. Now both of this pathway occur differently in different set of organisms, and what happens is they give a very similar intermediate. This intermediate includes your isopentyl pyrophosphate and dimethyl allyl pyrophosphate, and this is the basis for all the isoprene units. Now this isoprene units gives rise to lot of terpene derivatives. Now to understand terpene derivatives, let's just put some numbering to this isoprene so you can relate to it. Now I'm just putting numbers like 1, 2, 3 to call this as a head, and then 4, 5. I am numbering the carbon atoms. So here we have 5 carbon atoms representing the 5 carbon unit. Now if I see my first molecule, geranyl, geranyl is something which is giving you a nice rose like aroma. You have it in the rose, you have your geranyl, you have your geranyl acetate and so on. So I have picked up geranyl acetate as one of the molecule and see if you can correlate your isoprene unit here. Now I am just putting the numbers in a very same manner. 1, 2, 3, that's the head, and you have your 4 and 5. Just cut this molecule here. I can again restart 1, 2, 3, that's the head, and again 4, 5. Okay, and this moiety comes from your acetyl CoA. So this is a different unit, and here you have 2 isoprene units joined. This I call it as head and this I call it as tail. So joined in head to tail manner to each other, and these are called as condensation products. Now this may be polymeric in nature. Now take for example lycopene. Lycopene is a very red pigment which is present in your tomatoes. So if you see that red coloration of your tomatoes, now what happens, it's very big, and seeing the number of double bond, it absorbs in the UV region. So can I figure out my isoprene in that? Definitely yes. So I am just putting it randomly for you again. 1, 2, 3, 4, 5. Now you can correlate very easily. 1, 2, 3, 4, 5. Okay. Again cut it. 1, 2, 3, 4, 5. So you see this pattern being repeated almost 1, 3, 4, 5. Now what happens here is, now again I don't see a head, but if I just start from the reverse side. So let me start from here. 2, 3, 4, 5. Again 1, 2, 3, 4, 5. So here you can see a nice head to tail again. And I am cutting it here. 1, 2, 3, 4, 5. Now what happens in this case is, in most of the cases your 1, 2, 3 is joined to the 5, that is your head is joined to the tail. But when you talk or when you see this molecule here you see 5 is attached to 5. This is what is called as tail to tail condensation. So in molecules like lycopene, you will see 4 molecules joined head to tail, 4 molecules of isoprene joined head to tail, and this 4 plus 4, that is 4 5s are 20 plus 20 carbons are joined to each other by tail to tail condensation here, and that's where you have your C40 molecules, and that is how you can correctly draw the structure of this molecule. Now going to the next one, we have our next building block, that is your phenylpropenoids. You might have come across phenyl propenoid unit. Now this phenyl propenoid unit comes from building blocks such as phenylalanine and tyrosine. They are synthesized from pyruvic acid and erythrose 4-phosphate, which are coming from your Embden-Meyerhof as well as hexose monophosphate shunt respectively. Now this gives rise to shikimic acid and thereafter gives rise to phenylalanine and tyrosine. Now during this process they undergo deamination and are left with just phenyl and propane groups. So these are your phenyl propane derivatives. Now if you see phenyl propane derivatives, in nature there are abundant examples of phenyl propane derivatives. We just saw an example of eugenol where we said this methoxy is contributed by SAM, that is S-adenosyl methionine, especially the methyl group. But the other part of it is clearly a phenyl propane. So here you can see one phenyl propane. You can see phenyl propane in your coumarin. So you can imagine here again you have your phenyl and 1, 2, 3. What has happened is a cyclization. You can see phenyl propane in molecules such as lignans, which are dimers of phenyl propanes, and again you have a phenyl. This methoxy is contributed by SAM, and again you have your 1, 2, 3. Similarly in this case you have your 1, 2 and 3. So this lignans are nothing but dimers. Now what happens if they polymerize? So in plants you have something called as lignified tissue. What happens here is, imagine lot of your phenyl propenoids just joining to each other via either a ether linkage or a CC linkage. Now what happens is, this polymerization, you can see the monomer is still a phenyl propanoid. So all of this phenyl propanoid will fuse with each other to give rise to compound called as lignins. Now this lignin deposit with cellulose and make your tissues lignified. The lignification strengthens the stem, and as a result the bark or the wood is no longer soft due to lignin deposition, and you can easily identify lignin deposits by staining it with reagents such as fluoroglycinol and hydrochloric acid, which stains the lignin or the phenylpropanoid moiety in it. Now going to the other functionalities, apart from that, your phenylalanine and tyrosine in case of nitrogen elimination may undergo a carbon elimination. In that case, they are left with what are called as C6, C2, that is 1, 2, but instead of 3, here I am saying N. So it is a C6, C2N, or called as a phenylethylamine derivative. Now you will come across some phenylethylamine derivatives in nature which are hallucinogenic or extensively CNS stimulant or either analgesic in nature. An example of CNS stimulant is mescaline, which occurs in certain cactus species such as peyote, and a good example of analgesic agent is your alkaloid morphine. Now if you see, you can again see here the methoxy is nothing but contribution of SAM, but this is not a phenylpropanoid, this is a phenylethylamine. Whereas in this case it is a little difficult to visualize, but let us try. So here you have your phenyl. Let us put your carbon number 1, this is your 1, this is your 2, and this is your nitrogen. So you have your phenyl, this is 1, this is 2, and nitrogen, phenylethylamine, and again this methyl can be contributed by your S-adenosyl methionine. So in a similar manner, you can still have little longer functionalities, that is indole plus C2N. Now what happens, out of shigmic acid pathway you get one more amino acid which is tryptophan, and tryptophan contributes not only to the indole, that is your C6, C2N, but in addition to that you can still retain 2 carbon and a nitrogen. So imagine a decarboxylation happening, what I am getting is a indole plus a C2N. So indole plus C2N is, you can see a very big functionalities and which is present in certain alkaloids. Now these alkaloids are very big in their structure. To give you an example of such alkaloid which contains indole C2N or tryptophan unit, the two common ones are reserpine and vincristine. So you will see in this case, this is, I am just drawing it for you so that it is more convenient for you to visualize. So this is my indole, carbon number 1, carbon number 2, and N. So I have my C6, C2N, that is indole plus a C2N. Again, if you see a complex molecule such as your vinca alkaloids, vincristine, vinblastine, in same way they have the same indole moiety, and you can carefully see this is carbon 1, this is carbon 2, and N. So it's indole C2N. Not only that, because this is a dimer, there is one more place where you can localize this. One, 2, and N. So in vincristine it occurs, or the tryptophan moiety is substituted or added twice. Now you will go for more some nitrogen containing compounds which are also derived from amino acids. See, for example, you have amino acid ornithine. Suppose it undergoes decarboxylation and then deamination. When the moment it undergoes decarboxylation, it gets converted into compound called as putrescine. This putrescine will further undergo deamination and what will happen is when this amine is also lost, you are left with nitrogen and 1, 2, 3, 4. 4 carbons. So, I am just putting nitrogen plus 1, 2, 3, 4. That is, you get your pyrrolidine nuclease. Now, this pyrrolidine nuclease is involved in biosynthesis of numerous alkaloids, which include your hygrine, nicotine, especially this functionality of your nicotine, and atropine. In atropine, it is difficult to visualize, but let us try. So, this is your 1, this is your 2, this is your 3, this is your 4, and this is your 5-membered ring system. So, this is a fusion of 5-membered plus 6-membered ring system, wherein your 5-membered ring system is contributed by an amino acid ornithine. Now, instead of pyrrolidine, what happens if you want a piperidine ring? In that case, you have to increase the amino acid by 1 carbon, and that is the amino acid lysine. So, amino acid lysine is responsible for giving you piperidine kind of nucleus. So, amino acid lysine has 1, 2, 3, 4, 5 carbons and 2 nitrogens. Again, it will undergo decarboxylation to form what is called as cadaverine. Then it will undergo deamination and then cyclization to have all these 5 carbons. So, you will have 1, 2, 3, 4, 5 carbons and a nitrogen, which is your piperidine. Now, this piperidine is present in alkaloids such as basically if you see piperine, which is there in your black pepper, it is an amide alkaloid, but the piperidine ring is there. Then in your lobelia leaves, here you see, and annabacin, both the cases, as well as previously if you see the other ring of your nicotine, that is your pyridine ring of nicotine, that has also been contributed by your piperidine. So, these are all the building blocks that have been involved. So, just quickly going through it, we started with C1, that is your S-adenosyl methionine, then C2, which is brought in by acetyl CoA, then you have your C5, that is your isoprene unit, which is brought in by mevalonic acid or deoxyxylulose 5-phosphate. Then you have your phenylpropanoids, which have been given by your phenylalanine and tyrosine. Phenylalanine and tyrosine also give you what are called as phenylethylamines, which give rise to compounds such as your mescaline, morphine. After that, you have what are called as tryptophan derivatives, which are indole plus C2N derivatives, which are present in your reserpine, vincristine. Now, you have heterocyclic compounds, which are your pyrrolidine, and pyrrolidine is given by amino acid ornithine after decarboxylation and deamination, as well as amino acid lysine gives rise to piperidine. Now, these are few secondary metabolites, but these are not all. So, thank you everyone for your patient listening. Here are few references if you wish to go through the important parts or detailed discussion on how this biosynthetic precursor contribute to genesis of secondary metabolites. So, thank you everyone.